Hi everyone and welcome to today's video. This is a B1 level science lesson and we're going to be talking about data collection and analysis. So charts and graphs. What is the purpose of charts and graphs? Well, charts and graphs are a way of displaying the data or the results that you find in an experiment in a more understandable way. So we're going to read the two texts and then define the words below. So text A, scientists collect data and analyze the results of the experiment to determine whether the hypothesis is supported or refuted. If the hypothesis is supported by the data, scientists may refine their hypothesis to make it more accurate and complete. If the hypothesis is not supported by the data, scientists may revise or reject the hypothesis and develop new ones to explain what was observed. Text B, scientists do experiments to test their ideas about how things work. They collect information during the experiment and look at it to see if it agrees with what they thought would happen or if it doesn't agree. If the information agrees with their idea, they can make it even better. But if the information does not agree with their idea, they may edit their idea or throw it away and create a new one to explain what they observed. All right, so to support a hypothesis is to agree with what the scientists thought would happen. To refute a hypothesis is to disagree with what scientists thought would happen. To refine is to make even better. To revise is to edit. And to reject is to throw away. All right, now we're going to look at types of data. Most types of data can be put into two categories. So read each observation and decide if it is qualitative or quantitative. So qualitative data are observations using the five senses, before and after observations. Quantitative data are measurements of distance, weight, etc., number counts of items or trials, and estimates. So we'll use the blue for the qualitative and the orange for the quantitative. So qualitative data, number one, the nucleus is large and in the center of the cell. The liquid changed color from blue to green. It feels soft, and the mixture smells like a lemon. The quantitative data, the plant grew 0.5 inches from day 1 to day 4. The mass weighs 2.5 grams. The ball traveled 50 meters in 6 seconds, and the pH is 7. All right, these are some words and phrases that are common when collecting data. So try to match each word or phrase with its definition. You can pause the video, try that on your own, and then restart when you're ready to check your answers. Okay, so number 1 is A. Observations are information gained from your senses. Data is F. The information that is gotten from the experiment. Control group is D. A group that experiences normal conditions and is used to compare. Variables, C. things that can change during an experiment, such as temperature or time. Conclusions, G, the outcomes that are found after analyzing the data. Measurements are B, the size, length, or amount of something. And experimental group is E, a group that is exposed to the treatment being tested. All right, now you're going to complete each sentence with a word from below. Again, you can pause the video, try that on your own, and then restart when you're ready to check your answers. Okay, number 1, observations are important in science because they allow us to gather information about the world around us using our senses. Measurements give us information about the size, length, or amount of something we are testing. Variables can have a big effect on the results of an experiment, so it's important to control and measure them carefully. The control group is the group in an experiment that is used to compare. The experimental group is the group that is given the treatment being tested. Data collected during an experiment can be either qualitative or quantitative. And the conclusion is a summary of the results from an experiment and helps us understand what the results mean in the context of the experiment. All right, we're going to read a conversation between lab partners and then answer the questions. You can pause the video and read on your own, or you can follow along as I read. Hey, did you record the measurements we took earlier? Yes, I did. Did you get the temperature readings? No, I forgot to write that down. How much was it? It was around 25 degrees Celsius. Where do we record our findings? We can use the lab notebook. Should we use a table or a graph to represent the data? I think a table would be better because we have many measurements to compare. What do you think? Yeah, that makes sense. Let's create a table then. Do you want to do that? Sure, I'll work on that while you write down the procedures that we used. So, which words or phrases in the dialogue have the following meanings? So, to write down is to record. The data or measurements taken during the experiment would be the readings. The results are the findings. To change data into a more structured format is to represent. And to be understandable is to make sense. All right, so these are different kinds of graphs. Match each graph with its name. So pie chart would be letter B. Line graph is letter C. A bar graph is A. And a stacked bar graph is D. So match each graph with its use. Again, you can pause the video and try this on your own, and then restart when you're ready to check your answers. So a pie chart is B, useful for showing percentages or fractions of a whole, such as the percentage of people who are certain ages. A line graph is C. It's useful for showing changes over time or time trends in data, such as how the average amount of rain in one city varies throughout the year. Bar graphs, A, is useful for comparing values for different things, such as the average amount of rain in different cities. And a stacked bar graph is D. It's useful for showing the number of different parts within a whole or total. All right, so which graph should you use to organize the data listed? Again, you can try these on your own and then restart when you're ready to check your answers. All right, so daily plant growth would be a line graph. Monthly rainfall in 3 locations over a period of a year would be a stacked bar graph. The speed of different objects when falling, could use a bar graph for that. The effect of different temperatures on the growth of tomatoes, could use a line graph. The change of heart rate during a workout, line graph. The amount of time that different brands of batteries work in a flashlight, that would be a bar graph. The amount of rain each day in June in one city would be a line graph. And the average temperature in different cities in June, a bar graph. So independent and dependent variables. Read the experiment summaries below. What is the independent variable and what is the dependent variable? The independent variable is the variable that is changed by the researcher. The dependent variable is the variable that is measured or observed. For example, pretend that a researcher is conducting an experiment to determine the effect of temperature on bacteria growth. The independent variable is the temperature because this is controlled and changed by the researcher. The dependent variable is the bacteria growth, which changes as a result of the different temperatures. So different fertilizer is used on plants in a greenhouse for 2 months. The number of tomatoes on each plant is counted at the end of the 2 months. So the independent variable would be the fertilizer, and the dependent variable is the number of tomatoes on each plant. 4 plants are watered, each with a different amount of salt in the water and one with no salt at all. After 2 weeks, the height is measured. So the salt in the water is the independent variable, and the height is the dependent variable. One group of fish is fed a normal amount of food once a day. The second group is fed twice a day, and the third group is fed 4 times a day. The body fat of the fish is recorded each day. So the amount of food is the independent variable, and the body fat is the dependent variable. Creating graph titles. Science graphs often have titles with similar structure. Using the instructions, create titles for the graphs below. So you could have the effect of the independent variable on the dependent variable. Another structure could be the relationship between the independent variable and the independent variable, or the independent variable versus the dependent variable. So a ball is dropped from different heights above the floor, and the height that it bounces is measured. So for graph 1, the title would be the effective distance a ball drops on how high a ball bounces. Graph 2, a candle was burned under glass jars of different volumes to see if the volume of the jar affects the length of time that the candle burns. So the title would be the relationship between jar volume and the time a candle burns. And graph 3, geologists wanted to know if there was a relationship between the density of a rock and how many meters down it was collected from. So the title would be depth of collection versus rock density. All right, we're going to read the first part of another dialogue and then answer the questions. You can pause the video and read on your own, or you can follow along as I read. Hey, did you finish collecting the data for the science lab report? Not yet. I still need to measure the temperature and pH levels of the samples we collected. Okay, I finished measuring the mass and volume of the samples. Do you need any help with the temperature and pH measurements? That would be great. Do you know how to use the pH meter? Yeah, I used it in my chemistry class last semester. It's not too hard. We just need to calibrate it first and then dip it into the samples to get the pH readings. Awesome. Let's get started then. I'll take care of the temperature measurements while you work on the pH readings. So, what is the most likely meaning of the word calibrate? Well, that would be A, to check or adjust to make an accurate measurement. And why does Kevin know how to use a pH meter? He used it in another class. Now we'll read the second part of the dialogue and answer the questions. Okay, I finished measuring the pH of all the samples. They range from 6 to 8, which is within the normal range for fresh water. That's good to know. The temperature of the samples range from 20 to 25 degrees Celsius, which is also normal for fresh water. Great, we have all the data we need. Now we just need to input it into the lab report and analyze the results. Yeah, let's do that later today. Thanks for your help. No problem, anytime. Right, so which of the following temperatures could a sample have measured? So they said it was between 20 and 25 degrees. So C, 21 degrees would be a possible reading. And what do Amy and Kevin still need to do? They still need to put the data into their lab report and analyze it. So Amy and Kevin recorded the data below, and how can you put this data into a graph? And what can you learn from the data? Well, you could use a bar graph for each type of the fresh water, the tap water, the salt water. They could use possibly a line graph showing a different line for each of the types of water. Um, we noticed that before the rain, the fresh water had a lower pH than it did after the rain. The tap water was unchanged, obviously, because it's not affected by rain. And the salt water um, had also had a higher pH. Well, in the first instance, it had a higher pH, but in the next two, it had a lower pH. So those are some observations that you might be able to make from this data. And that brings us to the end of today's video. Please like, subscribe, share, and if you have any questions, leave those for me in the comments. Thanks for watching, and I'll see you next time. Bye.